So to start, as a background to my talk, in my thesis, I dissect the mechanisms underlying two distinct behaviors, sickness and foraging behaviors. And while these behaviors are very different from each other, what ties them together is that they're both long-lasting behaviors. And we're all familiar with long-lasting behaviors because we ourselves engage in them regularly. An evident example is that of sleep and wakefulness, where animals overtly appear to alternate between periods of rest and activity. Another example is that of sickness behaviors. So when we suffer an injury or an infection, there are a host of physiological changes that take place that are coordinated by the immune system as well as by the brain. But in addition to these physiological symptoms, our brain generates sickness behaviors, and these include reduced appetite, thirst, warmth-seeking behaviors, and more. And a third example is that of foraging behaviors, where we alternate between periods of exploitative feeding and exploratory food-seeking migrations. So in the field, long-lasting behaviors are often referred to as behavioral states. And I just want to make explicit what this terminology means as I use it throughout my talk. So just descriptively, these states outlast their eliciting stimulus. They do not map one-on-one to individual motor actions, but there is an overall behavioral pattern within these states. And importantly, they can last up to several minutes or hours. So a fundamental goal in neurobiology is to understand how the brain generates such behaviors. One mechanism that's considered to be pivotal for the generation of behavioral states is that of neuromodulation. So neuromodulatory pathways have been linked to many prominent behavioral states. For instance, the neuropeptide orexin, also known as hypocretin, projects from the lateral hypothalamus and promotes wakefulness behaviors. Second, in response to pathogen infection, the ADCY-AP1 neuropeptide was recently found to be released from a population of neurons in the mouse brainstem, and it's been linked to sickness behaviors such as lethargy and reduced appetite. And as a third example, the neuromodulator serotonin has been linked to many behaviors, including mood and arousal. But what seems to be conserved across the animal kingdom is that it promotes exploitative behaviors linked to feeding. So over here, when worms are placed on a plate of uniform bacteria, serotonin promotes exploitative behaviors and opposes exploratory food-seeking behaviors. And similarly, serotonin has been associated with exploitative feeding in zebrafish, as well as in foraging mice. So together, these examples highlight some of the neuromodulators that have been mapped onto distinct aspects of animal behavior. But what properties of neuromodulators allow them to underpin such behaviors? So first, I should make clear right out of the gate that neuromodulators are conserved signaling molecules. They're not unique to mammals and are expressed by both invertebrates and vertebrates. In terms of properties, neuromodulators, like typical neurotransmitters, can signal in a synaptic one-to-one manner between neurons. But they can also be released extrasynaptically and diffuse over large distances and signal beyond their synaptic partners in a broadcasting fashion. Another key property is that neuromodulators typically exert slow effects over long time scales. And this is in part mediated by them typically binding to metabotropic or G protein coupled receptors, whose activation then elicits biochemical signaling cascades that occur at slower time scales than, say, electrical signaling. So some neuromodulators, like the ones I've mentioned over here, have been functionally linked to behavior, but there are more than 100 neuromodulators that, for example, are produced in humans, and the function of a large number of these remain uncharacterized. Moreover, given our interest, it remains unknown whether they map onto distinct aspects of behavior. But even for the neuromodulators that have been linked to a specific behavior, the mechanistic underpinnings for most of them remain unclear. So to fully understand how neuromodulators drive behavior, we have to integrate multiple levels of analysis at the levels of genes, molecules, circuits and behaviors. And some questions would be to ask what ensembles of neurons known as neural circuits do neuromodulators act on? How do they change the function of these circuits? And how in turn are these neuromodulatory pathways themselves regulated to drive behaviors in a controlled manner? And establishing these links is not at all a trivial endeavor. So here's just to give you a sense of what we're up against while studying neuromodulatory pathways. For a lot of the neuromodulators, they're synthesized by neurons whose cell bodies are localized in these discrete nuclei in the brainstem or hypothalamus. I've represented this for serotonin, dopamine, and norepinephrine. And the cell bodies of these neurons integrate a complex array of inputs, top down and bottom up. But in addition, the projections of these neurons, as you can see over here, are widely distributed across the brain. And so the effects of their neuromodulators depends in part on the target cell types and the molecular and circuit properties of those target cells. To complicate this further, even within one target cell type, each neuromodulator can bind to many different receptors. So for instance, serotonin has up to 14 different receptors, and these different receptors can be co-expressed within the same cell types or cells. So the effect of the neuromodulator is contingent on the integration of these multiple receptors that it can bind to. And overlaid on top of this complex anatomy is the diffuse signaling property of neuromodulators, which further makes it hard to predict exactly where they're acting to exert their effects. So you can probably anticipate how complicated it can be to rigorously disentangle neuromodulatory pathways. And perhaps to crystallize this further, a famous scientist, Corey Bargman, described neuromodulators as the dark energy of the nervous system. They can be inferred, but not measured. So our approach to understanding how neuromodulators generate these long-lasting behaviors is to study it in a much simpler system where every neuron in the circuit can be identified, monitored, and manipulated. And by doing so, the goal is, first, we can establish mechanistic links at multiple levels of analysis in the simpler system, which is interesting and important in itself. And second, we can hopefully uncover conserved principles of how neuromodulators generate behavior. And the system we're using to study this is the roundworm C. elegans. So C. elegans are small worms, just a millimeter long. And you can see based on these numbers that it's much simpler than humans. But despite these overt differences, thanks to evolution, a lot of the core building blocks are present in C. elegans. So for example, in C. elegans, about 40% of the protein coding genes have mammalian homologs. Similarly, it has a rich behavioral repertoire like us humans. It senses touch, heat, odor, mates, rests, aggregates, and so on. And finally, many of the features of the nervous system of more complex organisms are found in C. elegans, as you can see over here. So in addition, several analyses and tools have been developed by the C. elegans scientific community that make C. elegans a really powerful model organism. So first, it has a fully mapped connectome. What this means is that the point-to-point physical connections between neurons are known, represented over here. Second, it has a great genetics toolkit, which allows us to target specific genes and neurons precisely. Third, it's optically transparent, which makes it relatively easy to visualize neural activity. So together, its biological features and the tools that have been developed for its studies, it allows us to perform analysis at multiple levels and develop an integrative view of how neuromodulators act on circuits to generate behavior. So now I'm going to dive into the behaviors I studied. In the wild, C. elegans feeds on a range of bacteria. Nutritive non-pathogenic bacteria are its source of food, but C. elegans also encounter pathogenic bacteria in the wild, which can cause inflammation, make them sick, and in some cases also cause death. So when worms are feeding on nutritive non-pathogenic bacteria, they engage in foraging behaviors, whereas when they feed on pathogenic bacteria, they show sickness behaviors. Other animals also display these behaviors, and overtly the specific motor actions within these behaviors are different and species specific, but the underlying behavioral states are likely to be conserved. And so my thesis uses molecular genetics and neural circuit perturbations to build on prior work and establish causal links between genes, neuromodulators, circuits, and behavior. And as I will show you, this reveals some principles about how these pathways generate behavior. So my thesis is conceptually divided into two parts. First, I focus on sickness behaviors and then on foraging behaviors. I'll spend most of my talk on the first part. So in the first part, I will characterize the hallmarks of sickness behaviors, and then I'll focus on the neural mechanisms that we've discovered to drive these behaviors. And I want to acknowledge my co-first author, Shriparna Pradhan, who has contributed equally to this study. For the second part on foraging behaviors, I'll just very briefly preview the work done here, some of which has been published. So now diving into the first part. The pathogen that we use to elicit sickness behaviors in C. elegans belongs to the gram-negative Pseudomonas genus. And actually, the specific strain we use is Ph.14, which was isolated a few decades ago from an immunocompromised patient. For the rest of my talk, I will refer to this specific pathogen by its strain name Ph.14. So Ph.14 is known to kill C. elegans using conserved virulence pathways. We know that one way through which Ph.14 kills C. elegans is through an infection-based slow killing process where it proliferates and colonizes the intestine. Based on others' work, we also know that C. elegans is initially attracted to the odors of Ph.14, but then on exposure, it avoids it. And additionally, we and others have observed that C. elegans that are feeding on Ph.14 begin to die around 2 days after infection, and all animals typically die within 5 days of infection. So first, we wanted to know what the different hallmarks of sickness behaviors are, and after that, understand how the brain controls these hallmarks. To begin, we characterize behaviors over the first day of infection before the onset of death. I just mentioned that death begins to occur on the second day of infection. And we started by looking at feeding behaviors, given they are particularly relevant for pathogen ingestion. And the setup for this experiment is shown here. We took wild-type animals and placed them on these plates which have bacteria on them. So either animals are placed on pathogenic bacteria, Ph.14, or as a control condition, the standard bacteria that we use in the lab would be P50, which is non-pathogenic. And after that, at defined time points, we measured animals' feeding behavior. So the feeding rate of uninfected animals placed on non-pathogenic bacteria is represented in black, whereas that of infected animals placed on Ph.14 is represented in bluish green. And on the y-axis, we have pumps per minute. So we're just quantifying the feeding rate. So pumps refers to the rhythmic feeding motions that occur in C. elegans. You can just think of it as a proxy for feeding rate. And what we find is that feeding rates in infected animals gradually decrease. So just to walk you through the first plot in detail, for each time point, we have the feeding rate for uninfected animals and then infected animals. And as you can see that at the start of their infection, these animals in bluish green have feeding rates similar to uninfected animals. But then as infection progresses, the feeding rate reduces. And we see these differences most reliably for later time points during the day. So next, we wanted to rigorously validate that these feeding changes were indeed due to infection. And for this, we first exposed animals to Ph.14 that was killed with UV radiation. So this bacteria cannot infect and colonize the intestine. After a day of being placed on these UV-killed bacteria, animals were transferred onto live pathogenic bacteria, and their feeding behaviors were immediately quantified. So despite acute exposure to this live pathogen, it appears that because these animals hadn't been infected, they had feeding rates that were similar to these uninfected animals on our standard food source. And with the animals that were exposed to live bacteria, as here, and then in all our standard cases, they displayed reduced feeding. So this suggests that only live Ph.14, which is capable of infecting C. elegans, can drive decreases in feeding. So quick checkpoint, on the first day of infections, animals progressively eat less. I don't have the time today to show you this data, but we found that after a day of infection, they also modestly decreased their speed, but did not change their reproductive behaviors or defecation rates. Strikingly, we observed that the feeding reductions from day one subsequently progressed into these short bouts of complete suppression of feeding. And this absence of feeding was often accompanied by an absence of movement also occurring in these short bouts. And in our field, when animals cease to eat or move for short bouts of time, this behavior has been described as quiescence. Just by way of background, C. elegans is known to display quiescence in 3 other contexts, not at all related to infection, during developmental larval transition, second in adults exposed to physiological stressors, such as UV or heat shock, and third, when adult animals become satiated on feeding highly nutritive bacteria. And the fact that quiescence behaviors have been described across these non-infected states becomes highly relevant in a couple of slides as we further dig into this behavior. So as a first step, we wanted to more thoroughly define the time course for these quiescence behaviors. So the experimental setup over here is similar to what I just described, except we characterize behaviors for multiple days until all infected animals have died. On the x-axis is the time point at which an observation was made. So for infected animals, it was 20 hours of infection, 96 hours of infection, and so on. On the y-axis is the fraction of animals that fall into one of 3 categories. Dead animals, animals that display quiescence, during which they do not move or feed for at least 20 seconds, and non-quiescent animals where they're alive and moving, but they're not showing quiescence. So first, I'll just show results for infected wild-type animals. As you can see, at the beginning of infection, all animals are non-quiescent. As infection progresses, this fraction decreases. And after more than a day of infection, some animals begin to show quiescence. After about 2 days of infection, animals begin to die. And this fraction of dead animals continues to increase until nearly all of them are dead by day 5. And to state the obvious, quiescent animals die too, which is why you see a decrease in the fraction quiescence for this category. And not surprisingly, when we did this same time course for non-infected wild-type animals based on non-pathogenic source of bacteria, we found that they did not display quiescence or death as they aged up to 5 days after being placed here as adults. And so this suggests that the quiescence and death observed at these time points and in these animals is specifically caused by infection. So as a quick summary of the first part, we characterize the hallmarks of sickness behaviors. Over the first day of infection, C. elegans eat less and move less. Subsequently, they not only eat and move less, but temporarily cease to feed or move in this bug-like fashion. And so they show quiescence behaviors. And then eventually, all infected animals die within 5 days, compared to uninfected animals, none of which die at this time point and are known to live up to 21 days. And so sickness behaviors such as decreased appetite and lethargy are conserved behaviors displayed by C. elegans and as we know in other more complex organisms. So now I'll be discussing the neural underpinnings of these sickness behaviors. As a start to dissect neural mechanisms underlying sickness behaviors, we focused on infection-induced feeding decreases. These are an early sign of behavioral changes caused by infection, and they're also amenable to rapid screening. So here is represented the feeding rate for wild-type animals at a single time point, which was the end of the first day of infection as the time point to assay, because this is when feeding rates of infected animals robustly decrease. We performed a behavioral screen on 19 mutant strains that lacked signaling pathways previously implicated in stress or pathogen-related behavioral changes. And I'd like to draw your eyes to a mutant that immediately struck out to us, the C17 mutants. So the C17 mutants were the only mutants that we screened that did not display these feeding decreases even after a day of infection. You see wild-type animals, they show these feeding decreases. C17 mutants do not. Infected C17 mutants do not. And in C17 mutants, the neuron ALA has impaired function. And as good geneticists, we first validated this phenotype by testing 2 other mutants. The mutant over here is original mutant. This mutant, which has a different loss of function allele, also has impaired ALA function, did not show decreases in feeding, and another mutant which independently impairs ALA function similarly phenocopies these others. So our genetics validate that the lack of feeding decreases in these mutants after a day of infection can be attributed to impaired function of the ALA neuron. So the ALA neuron regulating feeding decreases on infection was quite interesting because we know from the work of others that in an uninfected stress context, ALA promotes quiescence. And as I just showed you a couple slides ago, infection leads to quiescence too, but at later time points after these feeding decreases appear. I had mentioned earlier that quiescence behaviors are also known to be generated in 3 other infection states, and ALA is involved in one of them shown over here. So in this case, when animals are exposed to high temperature, then as a response, they display quiescence behaviors where their feeding and movement is suppressed. And ALA is known to promote quiescence, and this in part is mediated by the neuropeptides it is known to express. So promoting quiescence means suppressing behaviors. We found on infection, ALA is required to suppress the feeding behavior. So this function aligns with its known role in the stress state. An obvious next question is that do ALA-associated neuropeptides that suppress behaviors in the heat shock state also do so on infection? And we found this to be the case. So these 3 neuropeptide encoding genes known to be expressed by ALA, their mutants phenocopy the ALA defective mutant C17. However, another neuropeptide mutant, FLIP13, has a dramatically different phenotype. So unlike C17 and these other neuropeptide mutants, FLIP13 mutants did display feeding decreases on infection. But unlike wild-type animals, these feeding decreases were far more exaggerated. In fact, feeding in these mutants did not just decrease, but we observed these bouts of no feeding that were often accompanied by these bouts of no movement during infection. And we've seen that when these bouts of no feeding and no movement co-occur, then these we describe as quiescence. So we've seen quiescence in wild-type animals, but it only appears starting 2 days of infection, which is still another day away from this time point over here. And given this very striking early onset of quiescence in FLIP13 mutants, we wanted to characterize their sickness behaviors over longer time courses, as we had done with wild-type animals. So you've seen this setup. I'll go through this quickly. We place animals on pathogenic bacteria, categorize their behavior, and with FLIP13 mutants at the beginning of infection, all of them are non-quiescent. As infection progresses, this fraction decreases. And they show peak quiescence on just a day of infection. So in comparing to infected wild-type animals, it's quite evident that the onset of quiescence and death in FLIP13 mutants is accelerated. So quiescence appears within a day of infection in FLIP13 mutants, whereas only appears another day later in wild-type animals. Death occurs 3 to 4 days in FLIP13 and day 5 or later in wild-type. And I don't show this over here, but we always run controls with uninfected animals placed on non-pathogenic bacteria. And in these cases for both wild-type and FLIP13, we observe no quiescence. So to study this accelerated quiescence, we focused on a single time point, 20 hours, for the manipulations that I'm about to show you. And 20 hours, which is roughly 1 day of infection, is when FLIP13 animals show quiescence, but wild-type animals don't. So they can be differentiated from each other at this time point. And so this is how we're representing data. Just for one given time point, we do not see quiescence in wild-type animals at this stage. Whereas with FLIP13 mutants, about 30 to 50% of them display quiescence, and a smaller fraction display death. And again, we always run uninfected controls, but I'm not going to show those to you because we don't see quiescence. And quickly, just a few other experiments on the quiescence behavior in FLIP13. So first, as with wild-type animals, FLIP13 quiescence requires live PA14 infection. Second, the accelerated quiescence in FLIP13 mutants is not because they have deficits in pathogen avoidance behaviors. So basically, they spend a similar time being exposed to pathogenic bacteria as wild-type animals. And third, we tested 2 different stress pathways and found that these pathways are not dysregulated in FLIP13 mutants. So the accelerated quiescence cannot be attributed to a dysregulation in at least these 2 pathways. So to summarize these results, FLIP13 mutants display accelerated quiescence and death. What this suggests is that on infection, the FLIP13 neuropeptide delays the onset of quiescence and death. And this is striking because in the stress state, FLIP13 functions in the same pathway as ALA to promote quiescence, but on infection, it seems to have a different function from this ALA pathway that continues to suppress behavior. And instead, FLIP13 no longer promotes quiescence, but appears to delay its onset. So given the novel function of the FLIP13 neuropeptide that we had characterized in the infection state, we now wanted to map out the circuits that release this neuropeptide as well as its relevant targets. So first, to determine what neurons release FLIP13, we used a CRISPR-Cas9 genome editing strategy. To start here, this is the endogenous gene structure of FLIP13. In the mutant allele, the genetic mutant allele that we used, this region of the gene is deleted. And we generated an allele of FLIP13, which includes a T2A-M scarlet fluorescent reporter, as well as these LOXP sites. And the assumption is, and we found this to be true, that introducing this reporter sequence or these LOX sites at the regions that we did not disrupt native FLIP13 gene function. So the strain carrying this allele without any additional genetic perturbations should be like wild-type animals. So first, I'll just explain an experiment we did relying on the reporter. So the T2AM scarlet reporter was inserted before the stop codon, allowing us to visualize the gene's endogenous expression pattern. And this is an image of FLIP13M scarlet expression. Here we see FLIP13 is expressed in several neurons. I'd like to point out the ALA neuron and the I5 neurons, which seem to express FLIP13 at the highest levels. And next, we wanted to see if FLIP13 expression changed on infection. So we found that 20 hours of PA14 infection does not notably alter FLIP13 expression, either in the number of neurons that express FLIP13 or the expression levels within these sets of neurons. So next, we made use of the conditional knockout aspect of this allele. So here, the LOXP sites were placed before the second exon and after the last exon of this gene. So when we inject Cre recombinase into the strain, Cre would bind to these LOX sites and cause an excision. And this should delete the majority of the gene's coding sequence, as well as disrupt the M scarlet reporter. So the absence of the reporter is sort of a readout of the functional excision via Cre. And so you see this over here in cells that express Cre. You have disrupted gene function and no longer fluorescence, whereas in cells that do not express Cre, you should anticipate continued fluorescence and native FLIP13 function. So first, we validated whether this conditional knockout actually works. In the absence of the Cre recombinase, where FLIP13 function is intact, this strain displayed a wild-type-like phenotype, as you can see over here, which was encouraging. And on pan-neuronal Cre expression, where it should disrupt the expression of this neuropeptide in any neuron where it's endogenously expressed, we observed the same phenotype as in our genetic mutants over here. So in particular, these mutants display accelerated quiescence, and this was observed in 2 independent lines, and this provides additional genetic confirmation of the FLIP13 mutant phenotype. So based on publicly available RNA sequencing data, FLIP13 expression is detected in slightly more than 20 neurons. This is also, a lot of these neurons are also covered by our transcriptional reporter, and most of these are located in the head. In the literature, the ALA neuron has been associated as a prominent source of FLIP13 in the stress state. So first, we wanted to test if FLIP13 function in the ALA neuron is required for its roles in delaying the onset of quiescence on infection. And we find this not to be the case. So as you see, disrupting FLIP13 specifically from the ALA neuron does not lead to accelerated quiescence, and this strain phenocopies the wild-type-like control, suggesting that the FLIP13 release in ALA is not necessary for its functions on infection. So if not ALA, then which neuron is important for FLIP13 function? So instead of tackling each of these neurons at a time, we initially used fairly broad Cree drivers to identify if there was a specific neuron type where FLIP13 functions. And informed by these results, we can then use cell-specific promoters to drill down to single neurons. So here we find that multiple cell types each encompass neurons that are required for FLIP13 function. So disrupting FLIP13 in pharyngeal, sensory, and glutamatergic neurons each leads to accelerated quiescence and death, as we see with these FLIP13 global genetic mutants. So this maps our results onto known FLIP13 sources. Note that glutamatergic neurons encompass sensory as well as pharyngeal neurons. So now we wanted to test which of these specific neurons are required for infection-induced FLIP13 function. So I'm going to show you 10 different Cree strains, and I won't torture you with the details. But the takeaway here is that we find multiple neurons are required for FLIP13 function on infection. And neurons which seem to have a prominent function are the pharyngeal I5 and I1 neurons, and the sensory ASH and OLL neurons. So this suggests that on infection, a FLIP13 function is required in several specific pharyngeal and sensory neurons, but importantly, not in the ALA neuron. So we've discovered that FLIP13 function is mediated by a distinct set of neurons on pathogen infection compared to this other stress state. So using these same conditional knockouts that I just described, we did some converse experiments for the heat shock state, and I'll just tell you the takeaways. So first, we validated other studies and showed that indeed, ALA expression of FLIP13 is critical for its function in this state. And actually, we found that FLIP13 release from the I5 neuron, for instance, is not important for its functions in this state. And so together, what these studies really emphasize is that they're non-overlapping sets of FLIP13 releasing neurons that become important for its functions in the stress versus the infection state. We also mapped out what receptor FLIP13 acts on. And so FLIP13 is known to bind to the GI-coupled receptor DMSR1 to promote quiescence and heat shock stress. And we found that DMSR1 is also the relevant FLIP13 receptor on infection. So at this point, we've mapped out the infection-relevant neural sources of FLIP13, and that the FLIP13 functional receptor is DMSR1. But what about the quiescence behavior itself? So one core feature of quiescence that other papers have shown is reversibility. Which brings us to the question that during quiescence in infection, like during the stress state, are these also rapidly reversible? So to test whether quiescence is infection-induced quiescence is a reversible behavior, we introduced a transgene into the FLIP13 mutants. This transgene has been used in a previous study, and it allows us to acutely activate signaling in PDFR1 expressing neurons. And basically, these neurons are part of a neuromodulatory pathway that promote arousal-like roaming behaviors. So here, to acutely perturb these neurons, they were made to express a blue light activated adenylyl cyclase, which then activates signaling in these neurons and causes animals to engage in these aroused active moving states. So if quiescence was a comatose-like state, activation of this neuromodulatory pathway shouldn't be sufficient to rouse animals into moving or feeding. We find that this isn't the case. Quiescence can be rapidly reversed. So here, when we activate this neuromodulatory pathway, animals that were previously quiescent rapidly display high speeds. And in addition, we find that this activation can also cause animals to rapidly resume feeding at higher levels. So this shows that infection quiescence is not a comatose-like state, and in fact, it's reversible, where animals can resume engaging in vigorous movement or feeding after quiescence. Importantly, reversibility is a property that's also shared by quiescence caused by other contexts, for example, in the stress state. So now we've roughly mapped out the neural circuit pathway that delays quiescence and death onset. We've also done a bit of characterization on quiescence. And we find that in mutants lacking FLIP13 or DMSR1, there's this accelerated quiescence and death. But during these bouts of quiescence, when the animal is behaviorally very quiet, what's happening in the brain? Are there neurons that are actually active during bouts of quiescence? And if so, can they promote quiescence? Further, how do those neurons impinge on these neuromodulatory pathways? So in the final part of this section, I will describe an imaging approach we took to monitor neural activity across the brain during quiescence in infected FLIP13 mutants. To do so, we first crossed our FLIP13 worms into this transgenic strain where all neurons express GCAMP. GCAMP is a fluorescent molecule that becomes brighter when neurons have increased activity. And we have animals that express GCAMP, these fluorescent indicators, and we put these animals under a microscope that was custom built in our lab. And this microscope allows us to simultaneously record brain-wide neural activity with real-time behavior. And so this one that's placed over here, below it, there's a light path that allows us to record neural activity from the worm's head very rapidly. And above the animal, there's a low magnification setup that allows us to take images and quantify the behavior of the entire animal. And these images are analyzed in real time with a deep neural network that determines where the head of the worm is, where's the worm going, and so it automatically controls the microscope stage to move around with the animal. And if I hadn't emphasized it already, these are recordings in freely moving animals. That's what the point of this neural net is. And I'd like to emphasize that this is an incredibly powerful microscope and analysis pipeline that my colleagues in the lab have built. And I suggest you read a recent paper from our lab if you're particularly interested in this setup. So for our purposes, we infected FLIP13 mutants expressing GCAMP across the brain, and we placed quiescent animals under the microscope. So this is an example data set from one of the animals we recorded that enters and exits these short bouts of quiescence over an 8-minute recording. As you can see, during these quiescence bouts, the animals stop moving, feeding, and navigating. So what does the brain activity look like during quiescence? Below is a stack of neural activity traces from more than 100 neurons in the worm's head. These were all simultaneously recorded over 8 minutes. The color over here indicates the level of activity. So the brighter shades signify greater GCAMP fluorescence, which indicates greater neural activity, and darker shades suggest less activity. And you can observe that when quiescence bouts occur, a large fraction of the brain goes quiet. But in addition to these, we also observe a much smaller subset of neurons that display higher activity during quiescence. And so this was of particular interest because quiescence active neurons can also encompass neurons that promote quiescence. So now we have neural activity traces and we see how they relate to quiescence and non-quiescence bouts. But what is the identity of the neurons represented here, as well as in the additional traces we obtained from more animals? So for this, we took advantage of this transgenic strain called Neuropal that was developed by Oliver Hobart's lab. And this is a paradigm-changing tool in studying nervous system function. So in the Neuropal strain, a number of different fluorescent molecules are expressed in a stereotyped manner under defined genetic promoters. And this works because each neuron is genetically distinct, and it results in a fluorescent barcode for each neuron. So in worms, Neuropal serves as a landmark to assign unique identities to every neuron that is part of this dense volume of otherwise similarly labeled GCAMP-expressing neurons. So the FLIP13 mutants that we recorded here not only express GCAMP across the brain, but we also preemptively cross them into the neuropulse strain, so that once we obtain these neural activity traces, we could then map the identity of these neurons based on their position and then this multispectral atlas. And I should again point out that we were particularly interested in determining neurons that were active during quiescence. And so this entire setup essentially functions as a brain-wide imaging screen. So we were able to determine the identity of about 84 of these neurons. So these are all names of neurons across the 8 animals that we recorded from. And in this heat map over here, we quantify whether neurons elevate their activity during quiescence relative to non-quiescence. And this is indicated by hues of red, or whether neurons display less activity during quiescence, represented by hues of blue. And you can also see here white blocks. So the white blocks indicate that the neural identity of those neurons was not registered for that given animal, and this can be hard to do for a number of technical reasons. So white is essentially an absence of data. So first, I'd like to draw your attention to these neurons over here that displayed a significant decrease in activity during quiescence. And these included, not surprisingly, neurons that are known to encode locomotion and feeding. But we also mapped out the much smaller set of neurons that displayed greater activity during quiescence. For ESI neurons, for example, we observed elevated activity during quiescence bouts, and these are the neural activity traces from our brain-wide imaging recordings in 2 different animals. And we found that in both these animals, ESI neurons on average display greater activity during bouts of quiescence. And so this suggests that ESI are quiescence active neurons. So of the candidate neurons that we can validate, we immediately followed up on ESI because, as I'll describe later, it's been known to promote quiescence in a different uninfected state. So we were very excited by ESI showing up as a quiescence active neuron. To validate this result, we wanted to test whether ESI robustly increases its activity across many more animals. And for this, we used a much more rapid approach to image ESI, as brain-wide imaging is a relatively slower process. So for this setup, we constructed transgenic FLIP13 mutants that express GCAMP, but now not across the entire brain, only in the ESI neurons. And just like with our whole brain imaging setup, we infected animals, and then after a day of infection, we imaged quiescent animals. So quiescent animals move in and out of quiescence. So the Q bouts over here are when they are in quiescence, non-Q bouts are when they're not. And as evident here, ESI neurons on average display elevated activity during quiescence than during non-quiescence bouts. So as I mentioned, ESI was an especially interesting candidate that emerged from our brain-wide imaging screen. It's known to promote quiescence in this satiety-induced context. So here, animals that have fed on highly nutritive bacteria, or in case they're fasted and reintroduced to food, they become satiated and display satiety-induced quiescence, where there's a suppression of behaviors. And ESI promotes satiety-induced quiescence through its release of the DAF7 TGF-beta neuromodulator. And so this led us to ask, does this ESI DAF7 pathway also promote infection quiescence? And whether ESI is not just a quiescence active neuron, but also a quiescence promoting neuron. So for this, we needed to perform causal manipulations rather than just rely on imaging, which is simply correlational evidence. So these are the last sets of data for you. We found that disrupting ESI function through its genetic ablation suppresses quiescence in FLIP13 mutants. And similarly, disrupting one of its known outputs, TGF-beta, DAF7, also suppresses quiescence. And this suggests that ESI and DAF7 each are required for PA14-induced quiescence. Now, as you can see, disrupting DAF7 suppressed quiescence to a greater extent than ASI. And I won't show this to you, but we also tracked these infected animals over the longer time course assays that I've shown you earlier. And we found that ablating ASI did delay the onset of quiescence in FLIP13 animals, but DAF7 disruption had an even stronger effect, suggesting that there may be other DAF7 releasing neurons that promote quiescence. So these results add on to our model, which is that FLIP13 delays the onset of quiescence, and this in part is mediated by ASI and DAF7. So likely FLIP13 inhibits ASI. We haven't proven this directly, but our genetic experiments suggest this directionality. And in addition, since ASI ablation did not completely restore the onset of quiescence in FLIP13 mutants to wild type, it suggests that there may be other parallel pathways involved. And so I've represented that through these two separate arrows, suggesting that there's more than one quiescence promoting pathway. So this brings us to an overall summary. We identify neuromodulatory pathways that control sickness behaviors at different epochs of infection. A key principle that is revealed by this study is that unique internal states do not necessarily map in a one-to-one fashion onto unique sets of neuromodulators. Instead, what we see is that the function of the same neuromodulator can be repurposed across distinct states. And two findings illustrate this principle. First, we find that the neuropeptide FLIP13, which promotes quiescence in the stress state, instead delays quiescence on infection. So the same neuromodulator has distinct functions across distinct states. And we find that this functional switch is in part determined by which neurons release FLIP13. Second, we find that the DAF7 TGF-beta neuromodulator, which promotes quiescence in the satiety state, is recruited so it continues to do so in the infection state. And so neuromodulators can be promiscuous signaling molecules. They do not map onto singular states or singular functions, and they can be repurposed from distinct sources, as FLIP13 is, and in different combinations, as with TGF-beta, to specify unique internal states. So now, as I end this part of my talk, I'd like to highlight some mysteries that remain that we would like to further explore. I'm just going to highlight three for now. First, why does infection make only a specific set of FLIP13 releasing neurons important for its infection? Why is ALA no longer a relevant source of FLIP13 on infection? We found that the functional involvement does not map onto neurons that express FLIP13 at the highest levels. So another aspect to assess would be to determine what other properties are common to these neurons, such as what co-transmitters they might release with FLIP13. Second, what other pathways in addition to ASI promote quiescence? And so, as I've told you before, ASI ablation did not fully rescue the onset of FLIP13 quiescence to wild type, and so it suggests there may be other parallel pathways at play. And I think this is important to map out because the brain is quiet most of the brain is quiet during quiescence bouts. And so fully mapping out the neural circuits that promote quiescence should be useful. And third, we find that very different environmental triggers elicit the quiescence behavior. We know that the mechanisms underpinning quiescence are at least partially distinct across each of these states. So it would be interesting to explore whether the restorative or the homeostatic properties of the quiescence behavior also differ across each of these states. So now I will sprint through the last three slides that I have on the second part of my thesis, which focuses on foraging behaviors. So very quickly, switching gears, when C. elegans feed on nutritive, non-pathogenic bacteria, they alternate between two foraging behavioral states, exploitative dwelling and exploratory roaming. Roaming states are highly active states characterized by high speed and low turns, versus the dwelling states, which are these quieter states that include low speeds and frequent turns to densely sample within a small region. Now, these are locomotion states on foods, but animals are moving and feeding in both states. But as you can see, the overall behavioral patterns are quite different. Ethologically, Food ingestion cues are known to trigger the dwelling state, whereas chemosensory cues that can potentially signal the presence of better food or aversive stimuli promote the roaming states. So Niji, who was a postdoc in the lab, used circuit-wide imaging, and she found that the dwelling and roaming promoting neuromodulatory pathways opposed each other to generate these stable states. She also found that the activity of the AIA sensory processing neurons correlated with the activity of NSM, these dwelling promoting neurons during the dwelling state. But AIA's activity also correlated with the forward motion neurons during the roaming state. And this is where I came into this project. So my contributions make up the last two figures of this paper. And the experiments I performed provide causal evidence for AIA to couple sensory environments with either the roaming or the dwelling states. And I found that AIA activation can elicit both roaming and dwelling, and that it's necessary for each of these states depending on the sensory environment. And so what this suggests is that AIA is a flexible node in the switch between opposing stable foraging behaviors. And I also have some preliminary studies dissecting mechanisms through which internal satiety state may regulate foraging behaviors. So finally, to conclude, this thesis reveals mechanistic principles that generate long-lasting behaviors. With respect to foraging behaviors, we discovered a cellular node that allows animals to switch between stable opposing behaviors. And with respect to sickness behaviors, we discovered first how a neuropeptide can elicit different sickness behaviors based on which neurons release it and the environmental triggers. And second, we find a neuromodulator that drives similar behaviors despite distinct environmental triggers. And so my thesis reveals principles on how neuromodulators generate long-lasting behaviors at the levels of genes and neural circuit pathways. And this brings me to the end of my talk. So this work would not have been possible without my advisor, Steve, and very importantly, the numerous individuals whose names I have bolded over here. But really, every single person in the lab has enriched my experience through their scientific discussions, really thoughtful insights, whether during our meetings or outside informally. And I would like to thank my thesis committee. So Professor Soor is my committee chair. And one thing that I absolutely value about him as my committee member is that he asks deeply thoughtful and big picture questions that have been really helpful in my growth as a thinker. And personally, every time I interact with Mriganka, I'm just left so inspired by his enthusiasm for science and general optimism. So whether that was 6-ish years ago when I first talked to him during my PhD interviews here, or just a couple of weeks ago when I was stressing about my defense. I'd like to thank Professor Hurwitz, who, as many of you know, is a pioneering biologist and has been a leader in revealing the beauty of using C. elegans as the model organism it has become. And while I took 752 genetics, I was so often struck during Bob's lectures by the powerful and rigorous perspective that he used as a geneticist to approach scientific questions. And during my committee meetings, I've really valued Bob's curiosity, questions, and advice. And I'd like to thank my external committee member, Piali, and I've really valued Piali's questions and feedback, which have been useful in charting next steps with my science. And finally, my advisor, Steve. So it's been an absolute privilege to have undergone my scientific training with Steve, and that too as one of his very first set of graduate students. And Steve never fails to amaze me with his extraordinarily disciplined and rigorous approach to science. And it goes without saying that Steve is an exceptional scientist, but he's also a fantastic mentor. So his thoughtfulness towards all of us in the lab and his commitment to mentorship really help ensure that we bring our best to the lab and are also sufficiently happy when experiments or things are not working. And Steve's values really permeate into the overall lab environment, making it very collegial. So here's the rest of the lab. We all work hard, but also have fun times at socials and holiday parties. And I'd like to thank all my colleagues, past and present, some of whom are my very good friends, for their brilliance, the occasional misery pokers, but also all the fun times. And finally, I'd like to thank my friends and my family. My PhD would have been a very difficult journey had it not been for the times I spent with them. And many individuals over here, and also some whom I couldn't find photos with, have been a rock solid support system for me. And I'm very grateful for that. So thank you for your time and attention.